We're going to do this about talking about mercury toxicity because I think it's fundamental to the cause of many diseases. Even diseases that they say are caused by other divalent metal ions. I think these divalent metal ions are released from their binding sites, their normal binding sites, by exposure to mercury. And so we can talk about the sources of mercury, and some people say, well, is there any? And here's some publications, because I think it's easy to stand up here and wax eloquently and say things that you have no foundation for. I always like to have a scientific article or a report that supports what I'm doing. And this is the high level of mercury found in North Shore babies. This was in 2012. And what they found, it says we've never had hard evidence that there were exposures, said Pat McCann, the research scientist who conducted the study for the Department of Health and the Environmental Protection Agency. This study provides that. Yes, there are children in this country that have high mercury levels from a lot of different sources. The most prominent, in my opinion, is the thimerosal given in vaccines. This has now been eliminated, but back in the 2000 area. And the World Health Organization says, look, it's the major exposure. But I can tell you, you can get this much excess over seafood by 1 or 2 amalgam fillings if they're placed improperly or if they're in a chewing surface. So this is an exaggeration in the opposite. The actual level of dental amalgam exposure and presentation of mercury is quite remarkable. And this was a study done by Lisbon, Portugal. It was called the Children's Amalgam Trial, and they were doing this to show that amalgams were safe for all children. And this comes up, this is a fundamental thing produced by the government, people who absolutely hate me, and the IAOMT. And if you look at this, this is their study, and they separated it out. The first one they published it, they put the boys and girls together because they didn't want people seeing this type of data. Now what this slide shows is that boys excrete mercury at a much lower level than girls, and as they age, and if you go from the age of 7, which is right here, out to 14, what goes up? Testosterone levels. And as you increase the testosterone production in these children, the boys stop excreting mercury almost altogether at this time. And the girls maintain a high level of excretion. And I would point out to you that with autism and with attention deficit disorder and other autism spectrum disorders, boys vastly outnumber girls, for a minimum of 4 to 1, and in some diseases is 10 to 1. So why is that? Well, the difference is it's a risk factor. Testosterone is a risk factor for mercury exposure. It decreases your ability to excrete mercury, so it builds up in your body. And you have to answer this question: what happens to the mercury that the boys don't excrete? Where does it go? I mean, it's not going out in their urine, probably not in their feces either. And this is to me a very damning thing. And even looking at this evidence, the CDC and the FDA have no problem saying amalgams are safe for all children. I mean, you have to be somewhat demented or non-caring to look at data like this and not get concerned in light of the fact that most children that are affected with the autism spectrum disorders are boys. And we need to identify the causal agent, and the causal agent has to affect boys more than girls, and we got it right in our hands, and even our own government produces this type of data. And so we can say this especially affects boys, who are 4 times more likely to have autism or attention deficit hyperactive disorder than girls. And this is consistent with reports by the EPA and the National Academy of Sciences that 8 to 10% of American women have such high mercury body levels that would render increased susceptibility to neurological damage children they have. And I would point out there's other data that's been presented by another person who does not want to think that thimerosal causes autism. But what they showed in the amniotic fluid of mothers, this was done in London, Baron Cohen is the researcher's name, and when I first reported the effect of thimerosal and testosterone on neurological death, what they found is that I looked up this data, and they found that in the amniotic fluid of parents or women who give birth to autistic children, that they have very, very high testosterone levels relative to the controls. So testosterone is a risk factor for, testosterone levels is a risk factor for autism. And in our data, we get down to the biological level, you see that it increases the retention. Now, the thing that I think also I feel compelled to get everybody aware of is that there are HG emissions from anthropogenic sources worldwide, and this is shown in this slide. It's easy to see, and what we can understand if you look at this is that in China and in Korea and places like that where it's red shows a higher level of mercury that's being exposed, that the development of industries in these what used to be third world countries or countries that didn't have as much industry is really increasing the amount of mercury that's being released at different sites. And you can see the same thing happens in the United States. And if you look at this, this is called mercury that's coming over from the Mongolian flume. And the mercury from these countries spreads out all over the world, and all over the world, we've got a major problem. We have to decrease the use of mercury. And the United Nations has taken this up, and this is in sharp contrast to the American Dental Association says, no, put it in your mouth, it'll be safe there. So we have to be aware that global distribution and total wet and dry decomposition of mercury is really quite high. And we had a speaker here several years ago, Dr. Dan Lax from UCLA, who used the INHANE study. Again, a government-issued study. It's called the Nutritional Health and the National Health and Nutritional Evaluation Survey. And what he showed that 2% of the women aged 18 to 49 in 1999-2000 had levels that you could detect in the blood. And that level rose to 30% by 2005 and 2006. I have no idea what it is today, but what it's saying is that in American women, when you test their blood, they are getting an increased level of inorganic mercury into their system from some source. Now, that wasn't dental amalgams, but dental amalgams add to this, and we need to develop an attitude that we need to eliminate all forms of mercury exposure to humans. And I don't know how we can do this when you have a government that thinks they have a right to change data and hide sources of toxic exposures. And then taking another step on this is what I just suggested, maternal amalgams and dental fillings as a source of mercury exposure in developing fetuses and newborn. And even this group said, well, maybe it's not enough to cause a lot of toxicity, but that's when the mercury levels were low. This was done in 2008. It says the levels in the cord blood were significantly associated with the number of maternal amalgam fillings. And dental amalgam fillings in girls and women of reproductive age should be used with caution to avoid increased prenatal exposure. Where do we get to be so cautious? Why don't you say it's like saying drinking and driving is a bad thing, but so we should look at it cautiously when we drink and drive. No, you should not drink and drive. And we should not be putting mercury into the mouths of people that are going to be involved in producing new children at all. I mean, this is stupid to say, well, it's just a little bit of toxicity. We can live with it, because what we're finding, and we'll talk about that later, is that depending upon your genetics, it can be quite devastating, even the low levels of toxicity. And to take another step, because I'm speaking to dental people here, and some of you are new, is that we look at it at this type of study. This was in toxicology and applied pharmacology in 1997. And they said it was concluded that amalgam fillings are a significant source of AG in the saliva and feces of humans. Significant. Unless you're eating a lot of fish, it's the only source. I mean, dental amalgams are an aberration, and they should never be allowed, should never have been allowed. And the fact that our FDA puts off making a decision on this and looking at the science and being unable to recognize the science tells you we're not fighting with people who have a scientific disagreement with us. We're fighting with people that have a criminal attitude about protecting the income of certain industries. It's not, the science is overwhelming that mercury from dental amalgams is the major contributor to human mercury body burden. And this was one of those talked about it when I talked about the 17 micrograms from the World Health Organization. That's a vast underestimation. And this study was done, published in Archives of Environmental Health in 1994 from Sweden. And what they said is that fecal excretions range from 1 to 190 micrograms mercury per 24 hours. And adding to that is the synergistic toxin sulfur, which was from 4 to 97 micrograms per 24 hours. And this is from Dennerwald. So with regard to a Swedish middle-aged individual, the systemic uptake of mercury from the amalgams was on average predicted to be 12 micrograms per mercury per 24 hours. And the exposure. Now, exposure is just one part of the equation, and this is what I'm going to try and get across. Depending upon your genetics, you could get rid of that. Or if you have the wrong genetics, you cannot get rid of that, and it builds up in your body, and it causes you, by the time you reach 50 or 60, to have a high mercury body burden, a high load of mercury in your body that is quite capable of causing a lot of difference. Now on this one here, note the scale on this, because it's a lot different than what it shows, the level that goes up. And what this absolutely shows is that in the pituitary gland, the cerebral cortex, the thyroid, and the renal cortex, depending upon the number of mercury amalgams you have, the level of mercury goes up dramatically. And look at the error bars. They're quite remarkable. I mean, they're quite high. In other words, some people maintain a lot. If you look at the error bar on one of these, I'm sorry, I'm at an angle here where I can't see very well. But if you look at the very center ones, you can see that you could be down way below the average on having no amalgams. At the same time, you could go way above it when you look at the top error bar. And there are some people that just do not excrete it very well. And this is the one that really is makes me more angry than anything else. This is idiopathic dilated cardiomyopathy. This is a disease where you die of your heart just stops beating. Sudden cardiac death, they call it. And if you look at this, pardon me, I don't know why this is doing this, but you can look at this. The people who die of sudden cardiac death have 178,000 plus micrograms, nanograms of mercury per gram of heart tissue. That's an incredible amount. You've never seen that level in the blood of anybody. What this is telling you, in the people who are susceptible to this, usually young boy, and usually it's young male athletes that die, and the older people, it's the male who's out jogging when he's 55 and drops dead. And you have to say, how does that mercury get there, and where does it come from? That's a reasonable question. That's what we pay the NIH to find out. And they have refused to put a request out for research to determine what causes this high level of mercury in people. And there's 300,000 a year in the United States that die of this disease. This tells you that our government is doing everything it can. The health aspect of our government is doing everything it can to try to decrease the American people's knowledge of the importance of mercury toxicity and mercury exposure in our death. I mean, there's no other explanation for this. This is just unreasonable that they haven't gone out and found out where does this mercury come from? Because there's only one source where it could come from, to be honest with you, and that's dental amalgams. I'm going to have to tell you what this says, because somehow this transferring it from my Apple computer to here is quite different. And what this is showing is that mercury from dental amalgams and thimerosal from vaccines have a synergistic type of toxicity. In other words, the two together are much more toxic than the additive value of the two. And so a person that has a large number of amalgam fillings, like maybe a 55-year-old man going in to get his flu shot, when you expose him to mercury, he'll retain more than the person who has no amalgam fillings. And that is one thing that we know that from many scientific studies, is that the higher the level of mercury, the less you can excrete it. In other words, mercury inhibits its own excretion, so you have to be very, very careful with it, especially if you're being exposed to some extent. And I apologize for this slide jumping around like this, but what this is showing is that if you look at the numbers here, that's one of those is thimerosal, one of those is mercury alone. You put the two together and you see a much more rapid death of the neurons. And I also, at this time, I can point out that if you want to see a major change in the toxicity of thimerosal, add testosterone to the neurons in culture. It's astounding. And I don't know why the government does not pay somebody a large amount of money to do a study on this issue. So let's talk about what are the primary chemical actions, because I like abusing some people here and making them learn a little bit of chemistry. The chemical actions of mercury are much more diverse and much more critical than what almost all the toxicological tests and the people who are experts in toxicology will tell you. We know it covalently binds to the SH groups, the thyroid groups of biomolecules, and inhibits their activity. In other words, it'll inhibit the enzymes that make glutathione, which you need for getting other toxins out of your body. But this is the one you have to keep in your mind. Mercury, when it gets into your body, displaces other essential metals. In other words, you have enzymes that require zinc, copper, and iron that are essential metals. They do essential things. Mercury, if you have an iron-binding protein and you put mercury in that, and that mercury gets converted to Hg2+, it will displace that iron, and two things happen then. You inhibit that iron, catalyze that iron-requiring enzyme, and you replace iron, and free iron in the body is extremely toxic. If you read the current literature today, the people who are working on Alzheimer's disease, Parkinson's disease, and about every other disease are saying that the metals that are involved are not mercury. They're iron, copper, and manganese. However, how do you get those metals in your brain? Iron cannot go through your body just because of just think of all the citric acid it would have to pass. How do you get iron in your brain? Well, you have iron goes there by a natural carrier protein. It gets placed properly in the body, but you breathe mercury vapor. Mercury vapor immediately can pass through the blood-brain barrier, get into the brain, gets oxidized from Hg0 to Hg2+, and then it displaces the iron in the electron transport system. You can't make ATP. You can't make glutathione. And then you release that iron, and that iron starts taking the oxygen that you breathe to make ATP. It starts converting that into superoxide anion, and the next pathway down there in the Fenton reaction it gets converted to hydroxy free radicals, and you suffer from oxidative stress. So this is a complex thing, and it's kind of said here in the bottom here, displaced redox active essential metals like iron and copper are proposed to be involved as electron donors in production of hydroxy free radicals and the symptoms associated with multiple diseases. But it's probably something like mercury or cadmium that's displacing these. Otherwise, you wouldn't have this problem because you can't eat too much iron. Your body won't let it come across your gut. Okay. So if we talk about one of the major biochemical actions of mercury toxicity, I want to bring in another thing here. It inhibits the activity of the enzymes that we talked about. It activates some destructive enzymes. Phospholipases, MMP, matrix metalloproteinase, and this leads to leaky gut. In other words, when you talk about how do bacteria and how do people develop food allergies, the only way you can develop a food allergy, and everybody said, well, your immune system went bad. No, it didn't. It did exactly what it's supposed to do. But if you take the mercurocell or mercury, all of a sudden you activate enzymes that chew up the collagen that holds your cells together in tight junctions. You destroy the tight junctions, and then you start getting leakiness. And then when the food you eat, such as gluten, that's still a peptide, should never go through your intestine. It should never get into your bloodstream. When it does get into your bloodstream, what happens? Some of the unusual peptides, your immune system does what it's supposed to do. It's a foreign body. It makes antibodies to it. So when you're getting your self-tested for food allergies, they call them, or adverse effect to eating certain foods, it's because you have a leaky gut. Because those should never, ever get in your bloodstream, should never be associated with your immune system at all. And when you do that, they say you have an immune problem. No, you have a leaky gut problem, or you have a leaky arterial problem. And I would think if you're a medical doctor and you think about people with thyroid problems, etc., every organ in the body has a lipid bilayer or a cell membrane that surrounds it that keeps it compartmentalized away from the immune system. If you lose that compartmentalization because you develop leaky guts, and I'm going to show you data that the mercurocell and mercury absolutely are critical and very effective in making all your membranes leaky. It makes your blood-brain barrier leaky. So this is the one that I'm going to try and make a point on here when we get a little further in here. And we talked about the disruption of the homeostasis of essential metals. I mean, free iron in your body. I mean, you've heard of iron overload disease and Wilson's disease with copper. When you start getting free metal ions such as these that are essential metals, you have to eat them. And you have to say, how do they get disrupted? What would disrupt? If you want to design a disruptor of iron or copper binding, what would you make? You'd make a metal ion that would bind tighter to those sites than the parent metal, I mean the iron or the copper. And trust me, mercury and cadmium will do that. And the other thing about the mercury toxicity, which all of us know about, is, and you have to ask the question, that mercury greatly enhances the production of toxic hydroxyl free radicals by the mitochondria, electron transport system. Mercury cannot be an electron donor. It's not a redox enzyme. In other words, it does not donate electrons to other sources. What it does, it displaces iron and copper, and they do the donation. That's where the oxidative stress comes from. And when you take mercury, I don't know how many hundred papers I've read that have shown that mercury causes increased oxidative stress in the brain. And yet it cannot itself, in test tube, make hydroxy-free radicals like copper and iron can. Well, then you have to say, well, it must be displacing it. And that's really one of the critical things. When people start saying, you know, Alzheimer's disease is caused by free copper in the brain interacting with beta amyloid protein, producing hydroxy-free radicals, well, how did the free copper get there? I mean, your body has tremendous defense against free copper getting into your brain. And so it has to get there by a natural carrier protein and placed in the body normally, and then mercury comes and displaces it. Essential biochemical systems, when I call it an essential biochemical system, that's a biochemical system that's found in even the lowest order of cell life. I mean, in like, a bug or a bacteria. And all semi-permeable lipid bilayer membranes that allow for selective compartmentalization of life-essential materials in the cells and excludes toxic materials become leaky and allow for destructive processes such as abnormal immune responses whenever you add mercury. ATP or energy-producing systems such as glycolysis and mitochondrial electron transport systems are inhibited by mercury binding, and that's a very basic thing. If you don't have that, you can't. No cell system can live without having an ATP or energy-producing system. And the univalent transport mechanisms, such things as a sodium-potassium ATPase, that's where when you talk about voltage being involved in your health, it's not by pH. pH inside your cells and pH outside your cells is different, but it's not enough to cause a big energy gradient unless you just talk about the mitochondria. But sodium and potassium pumped by ATP, that's what controls the energy flux of your cell or your membrane potential. And this is something that's dramatically inhibited by mercury. Mercury is a very, very important inhibitor of the sodium-potassium ATPase that translocates and makes a membrane potential, which is essential for you to take up amino acids, etc. And the last one I'm going to talk about is the reducing system that keeps the intercellular space in a reduced state. I mean, when we die, we start oxidizing. Death is oxidation. And the reducing system inside our body, the glutathione levels and the levels of other reducing equivalents such as NADH, etc., is where a big portion of our body's energy goes. And mercury totally disrupts that. If you see someone that's mercury toxic, they lack all sorts of energy. They can't take care of themselves. And I know some of you in here have been in that condition. So then we have to take a step back and talk about the evolved biochemical systems that allow complex mammalian life are negatively affected by mercury. The hormone-activated systems that respond to flight or fight, maintenance of pregnancy, basic metabolism, and thyroid gland. We'll talk about that one. I have seen, we know from research that mercury dramatically affects the production of thyroid hormone that's needed. And that can be reversed. And the problem there is that you make the membrane that surrounds the thyroid gland leaky, and it cannot survive that as well and make thyroid that's essential for maintenance of life. And the immune system that protects the body from microbial invasion has to be tightly compartmentalized. And this is the real take-home lesson. I'll say it several times so I know you don't miss it. if your immune system is not tightly compartmentalized, you're going to have all sorts of problems. Because when the immune system sees a foreign body, what does it do? You know, you have the macrophage cells that go up to that foreign body, they bind to it, and they engulf it, and then what do? They produce a huge amount of hydroxy-free radicals, because the hydroxy-free radicals will kill anything living that's a normal life, and they release a lot of hydroxy radicals that kill what they perceive as the foreign body. Now, if that foreign body happens to be a piece of food that gets into your gut or into your bloodstream that shouldn't get there, it's not really going to bother you very much, except that your immune system is going to attack it and going to increase the oxidative stress and kill other cells in your body, in your brain, in your blood system. Okay, we say this without a doubt: biomembranes that shield the various organs from toxic materials and the compartmentalization of the immune system are greatly damaged by mercury. It's one of the first things that they hit. When you breathe mercury, you get mercury, especially if you ingest mercury Hg2+, you're going to tear your membranes, your endothelial cells of your arteries apart. Your gut, you're going to develop leaky gut, and then from there, the immune system will do more damage to you. Instead of helping you, it starts hurting you. Okay, understanding membrane definitions is one of the first things, and there are two types of membranes we're talking about. The first is the lipid bilayer that surrounds each individual cell. And the reason mercury is so damn toxic is it goes through these membranes with a great deal of ease, and then once it's inside, it's like a terrorist. It gets inside, it gets oxidized to Hg2+, and it can then do a disaster to the entire system. And second, there are membranes, such as the membranes, the epithelial membranes that line your gut. They protect your immune system, and they protect your body from having things get across from from what you've eaten or drank into the body when they shouldn't get through. They limit the amount of iron that gets in your body. They limit the amount of copper and manganese. And so you have to have a system like this that tightly regulates how much metals are allowed into your body, and that's done through a tight membrane system, and we'll talk about that. But that's a system of cells attached to cells. And what mercury does, mercury activates an enzyme that's located in those membranes to help you repair yourself in case you have an injury. It's called matrix metalloproteinase. And when you activate that with mercury, and we proved that, I presented this to the Academy a long time ago, you add mercury to MMP and you activate it. And without mercury, it won't chew up collagen. You put a little mercury in there, and all of a sudden that enzyme chews up the collagen, and that's the connective tissue between your cells that gives you some compartmentalization. And this is exactly what we're talking about, and I'll try and give you. So you can see this is a single cell, and this cell is surrounded by a lipid bilayer. But cells like this, when they make tight junctions and line up such as this, will make a membrane that can surround an organ, and every organ is of any importance in the body has a membrane that surrounds it to keep the immune system away from it. And what we see is the matrix metalloproteinase is located in this type of region in between the cells, and it's inactive. It just sits there. When you break the cells, there's a protease that supposedly comes out, clips that MMP, and activates it so it can chew up the broken area, and you can rebuild it. That's how you start repairing your body. If you had mercury, without these cells being broken, you can see that this matrix metalloproteinase chews this up, and the cells start falling apart, become leaky, and they also undergo oxidative stress because some of the mercury goes inside and activates redox systems inside that lead to destruction, mainly by releasing iron and copper from their natural binding sites. And we're going to show you some of this data. This is done, I did this with Dr. Perinati at Ohio State University. But the concept, and I think it's important that doctors follow this, is compartmentalization. Your body has a thyroid gland, has an adrenal gland, has numerous glands, includes your digestive tract. And they have to be separated from the immune system and from each other. Otherwise, it would be a disaster. And to get things across, you have to have a carrier that is membrane soluble. That's how the body solves this. It allows iron to get across the gut only when it's bound to a carrier. That limits the amount of iron, and it brings it in without the iron being able to form hydroxy-free radicals while it's going through the body. Same thing with copper and manganese and other things. For example, iron and manganese have to be bound by a specific carrier protein, ferritin, for example, iron, before they can cross the epithelial membrane of the intestine. If you start knocking holes in it, then you can't regulate that. And we've shown that mercury, methyl mercury, and thimerosal have been destroy such tight membrane structures, causing leakiness and enabling the entry of toxic materials. And to sum this up, do you remember the old song when some of us were young? Some of you, it's "Ain't you glad you got skin?" Well, if you understand that concept, why you have skin is to keep crap from getting into your body. And but it goes a lot deeper than that. You have a type of skin around your thyroid gland, around your adrenal gland, etc. When you just start destroying that, then the integrity of the body gets down, and you can end up with multiple problems that today are not identified with heavy metal toxicity. Most medical doctors, I tell you, I've been sick recently, and I've had the misfortune of having to deal with some of your colleagues. And with the first speaker here talked about this. Every damn one of them, without doing one measurement, told me, "You need to be on a statin." I refused to take it. I mean, I know the papers research. I'm going to say he was right on when he talked about what they do to you. So we have a problem, and they're miseducated. They're not poorly educated. They're miseducated in medical school about what is important to people, and they don't have a clue. I'm going to tell you, I talked to them and asked them, because I'm generally interested in what they're doing. They don't have a clue about heavy metal toxicity. They don't have a clue about synergistic toxicities of iron and lead and mercury. They don't have a clue. So this academy has a big job ahead of it. You guys are going to be 5,000 before it's over, and it's going to take that many of you to get these people to work. And so we talk about the biomembranes, and I like saying they're the first line of defense. And the epithelial cell in your gut is the first one that most things run into. And then you have the aortic endothelial cell membranes. And we've done some research on this, looking at how can we keep and make these membranes better. And many of you know I was involved in a compound called OSR that I made primarily to help children. And invariably, what the mothers would tell me, the net effect of the compound primarily was that it improved their intestinal dysbiosis. And I think that's a good reason for using these things. And so when we talk about the collateral damage is done by mercury toxicity biomembranes, I mean, if you look at some, like we talked about the adverse food response or food allergies, you can't have that unless food's leaking into your bloodstream. I mean, it can't happen. So if they start teaching you and telling you, well, don't eat this, well, try to do something to prevent the leakiness of your intestine. And I think we've talked about this, and I'm going to try and get a little further on here. When we talk about iron, manganese, copper, and zinc, they're limited in their uptake by the body because they're toxic if they're in a free state. And what this particular slide is about, it's important to remember that essential minerals such as copper, iron, in the free states can induce severe oxidative stress due to their electron donating capacity and the hydroxyl free radical producing Fenton reaction. And that's exactly what when they're bound to their normal enzymes, they don't do that. Once they're released, displaced, and the only thing that I know of that can do it, although there are probably other chemicals that can do this, is to have them displaced by mercury. Okay. The blockage of capillaries leads to several systemic illnesses that have reduced blood flow and abnormal blood pressure as symptoms. And the release of material from the atherosclerotic plaques leads to increased immune responses to material identified as foreign. This site is well known to harbor infectious microbes from periodontal infections. And these infections go there because there's a lot of food for them to eat. I mean, a bacteria, which we talked about many years ago quite a bit, from periodontal disease or cavitations, once it gets in the bloodstream, can actually be taken care of by your immune system, but not if it can find a leaky site. If it can find a site such as an atherosclerotic plaque, and this is proven, the bacteria will collect in the atherosclerotic plaque reproduce and produce toxins at that site. There's no other way around it. It would do the same thing in your intestine. But so we have to look at these issues, and this may be the reason why root canal teeth and cavitations, as well as mercury, is associated with cardiovascular disease. And this is another paper that had a good conclusion. And this was inflammation, heat shock proteins, and periodontal pathogens in atherosclerosis. And it was an immunological study, histological study. And what they concluded with, these results give evidence for specific immune response associated with atherosclerosis, where the bacteria initiate the observed inflammation in atherosclerotic lesions is not clear. However, the present study shows that the maintenance of inflammation may be enhanced by the presence of periodontal bacteria. And what this says, once your bloodstream becomes leaky and you're leaking bacteria, microbes, from your root canal tooth into your bloodstream, if it doesn't have a place to harbor and dock like a leaky membrane, your immune system's going to kill it. But if you're a person that's taking mercury on a regular basis, getting a shot, and you cause lesions to form, these periodontal bacteria are going to set up house there, and then you're going to get worse. So the major concept here is primary mercury reactions leading to enzyme abnormalities further leads to the destruction of the integrity of cells making up the biomembrane. And the loss of membrane integrity is the main reason most people get severely sick. And this is, if you read the papers, just start thinking about some of the diseases that you know about. I mean, why do you have such a tremendous immune response? And I know many of you have gone to doctors that have drawn your blood and sent it off to laboratories and had it checked for leakiness. I mean, you see a lot of normal proteins that are in certain cells, such as antibodies against some of the tissues found in the thyroid gland are commonly found. And the destruction, and the whole cause of this is that you've lost your membrane integrity. Okay, we're going to talk about this, and I think I can do it better with a slide. How does mercury and certain other compounds affect this, and why do we need to address this? And this is a graphic of a cell membrane, and it's easily penetrated by mercury and organic mercury. For those of you that don't know, mercury vapor would much rather be in oil than in water. It actually hates oxygen. It will be repelled by oxygen, so it likes going into your mercury, into your lipid bilayer. And we show this here by showing that the mercury goes straight through the membrane, and it's only when it gets in here it gets converted to Hg2+, and that's the toxic form. Hg0 is not toxic. The ADA likes to make a big deal of that. The mercury in your dental amalgam is not reactive, it's not toxic. No, but it's the transport process to get mercury into the cell easier. And mercury vapor is much more toxic to your central nervous system than is Hg2+. Hg2+ won't cross the blood-brain barrier very effectively. But Hg0 goes in with a great deal of ease. If you expose someone to mercury vapor, it'll collect in the brain, and after it's in the brain, it gets converted to by an enzyme, some enzyme, but one called catalase, to Hg2+. And when it does this, it interacts in the mitochondria. And we're going to show you some data that it actually builds up in the mitochondria at a very high level, 1,000 times the level that you find in the blood. And when it does this, it shuts off your ability to make ATP, and your ability, and it allows the oxygen not to be used that you're breathing to make ATP, and that oxygen is what is used to generate hydroxy-free radicals. Now let's talk about some of the things. I know a lot of you in here, and I'll hurt some feelings here, but EDTA, DMPS, and DMSA do not effectively cross the blood-brain barrier. The mercury that's causing trouble is in here. It's in the cell, and you can't get in the cell with these compounds, at least not very effectively. Methylmercury and ethylmercury from vaccines are shown here. They actually exist in the body, and with the high chloride content, instead of being MEHG+, it's MEHGCl. That means it's neutral, and it can go through the membrane. When it gets over here, it can lose that chloride, and you can build this up inside the cell. It builds up. The mercury in the cytoplasm of a cell is 5.6 times higher than it is in the bloodstream. And And then when it gets in the bloodstream going into here, it goes up 1,000 times higher in the mitochondria. And this is all based on the membrane potential. You put something in that has a plus charge, and inside the membrane, like the inside this membrane, is much more negative than the outside. That's how you get a membrane potential, is the differential between negative charges on the surface, on the outside, versus on the inside. It's much more negative on the inside. So once this comes in, it's got a positive charge, it stays inside here because the positive charges do not prevent it from going out. Same thing with ethyl mercury. And the membrane potential of this is not that big. But with the mitochondria, it's 185 millivolts, so it really concentrates the mercury. Once it gets inside your cell, it gets concentrated in your mitochondria. And you've all heard about the people who have mitochondrial dysfunction. Well, I'm telling you, you load them up with mercury and they'll be dysfunctional. They don't try to identify what causes that. They can't find a gene that does it. And so what we worked on, and to give you just a little bit of an advertisement, and I'm telling you, I'm working on this drug. It's called NBMI in our parlance, and it goes through the membrane because it's not charged. It's also made of 2 dietary ingredients, and it binds mercury much tighter than any of these chelators here. And when it binds mercury, it can get out because it's a substrate for the P450 system that attaches probably a glutathione molecule to the entire mercury NBMI complex and carries it out slowly. But while it's bound, it's not toxic and it won't let it go free. We'll talk more about that later. Yeah, well, this is this thought, what we just talked about here, the fact that the inside of your cells are much more negative than the outside membrane. And when you have something that's positively charged, especially something that can go back and forth, like methyl mercury plus to methyl mercury chloride, and you have a lot of chloride in your blood, and so most of the mercury in the body has chloride bound to it. And when it does that, it can go right through the membrane, and then it concentrates on the side of the membrane that has the highest level of negative charges. And your body is designed to have more negative charges on the inner surface of the cell membrane than the outer surface. And when you get to the mitochondria, that's exaggerated even much more. So it leads to a concentration of mercury into the mitochondria. Okay, the answer is yes. This stuff does effectively cross the cell membrane. And I want to tell you how we know that. Mercury, and especially you saw this in children that were given vaccines with the Marisol, is that they have what they call the urinary porphyrin profile. And they can tell whether or not you're mercury exposed. And this is work done by many scientists, but most recently James Woods. And they look at the urinary porphyrin profile in your, and they can tell where it's blocked. And it's blocked right here. And this is on the outer aspect of the inner membrane of the mitochondria. That's where mercury blocks the production of heme and or porphyrins. And porphyrins lead to heme. If you don't have heme, you're in big trouble. Heme is used in a lot of different enzymes. Most noticeable in the ability to carry oxygen in your hemoglobin. And you've seen autistic children, they're kind of pale. Mercury toxic people are kind of pale. They're not making hemoglobin. It's one thing, you've got to be suppressed if you have a lot of mercury in your body. And that tells you the mercury gets in there without any problem. So the point I'm trying to make is since chronic mercury exposure inhibits the porphyrin pathway at the site of the inner mitochondrial membrane, we know mercury gets there based on the urinary porphyrin profile. It's a simple test. Science proves this. You can look at somebody's porphyrin profile and tell if they're toxic to mercury. You can also tell if they're toxic to lead because it interacts at a different site. And I would maintain that if you can reverse this with a compound, your compound's getting in the mitochondria. And we reversed this on a lot of people when it was readily given. The porphyrin profile would straighten up on the chelation of mercury with a proper deal. And this is the mitochondria, and again, just to show you, this is the site where the urinary porphyrin profile where mercury inhibits is, right? It's on the outer aspect of the inner membrane of the mitochondria. And this is where the electron transport system is found inside here. And the TCA cycles in the membrane inside here, and the TCA cycles in this loose space in between the membrane. And mercury gets in there, and what does it do? It inhibits a pyruvate dehydrogenase complex, which is part of the TCA cycle, where the pyruvate is produced. And as well as that, it inhibits the ATPase, or the electron transport, and you don't get ATP going out. and you end up with a lot of problems. However, the electron transport, everyone makes a little bit of extra hydroxy free radicals. We all have a level of those that are wasted. And when they go out, if you have a healthy glutathione system out here, you scavenge them and they cause no damage. However, if you put mercury inside there, the amount of hydroxy free radicals that are produced that they're showing going out of the mitochondria is dramatically increased, and your body doesn't have enough glutathione to take care of it. And if it does, it consumes the glutathione down to where you cannot detox other toxic materials. Because glutathione, in addition to being an antioxidant, is also the molecule that's attached to many, such as acetaminophen, Tylenol. When you take Tylenol, you can deplete your body of glutathione and to the point where you can die. That's because glutathione is attached to an adduct of a Tylenol that's toxic, and it is used to carry it out of the body. When glutathione is attached to a molecule, that molecule is marked for transport into the blood system and for excretion into the feces. If it binds mercury, it does the same thing. So this is a study, and the guys are here, and I want. The reason this study came out, modification of neural behavior effects by mercury by genetic polymorphism of porphyrin oxidation children. If all of you remember, we showed you that study where the amount of mercury excreted in the urine of boys was much less than girls. That was part of the children's amalgam trial. Well, this was part of the children's amalgam trial also. And what they showed was that putting in amalgam fillings didn't affect the urinary porphyrin profile. And I talked to the guys, and we had a discussion. That's totally different than what you find in dentists. I mean, it affects the dentists, so why wouldn't it affect in children? And this academy funded some research that the guys did to show that the data wasn't right. And what they did in that study, the first study, was that they took all the people who were in mercury to get amalgam fillings, even if they got none or one or two, and they piled them up with all those that got 15. And it was the ones that got the higher levels that started showing the effect. But you could lose the statistical significance by piling them all together. That's not even good science. And the guys that published this paper showing that, indeed, and this led Dr. Woods, I think, to start looking at the data a little more carefully. And what he found is that the ones that were affected with the high levels of amalgam fillings were those that had the CPOX4 gene forms that made them susceptible to mercury toxicity. In other words, it inhibited that enzyme that we just talked about on the wall of the mitochondria. So, but it's interesting, and among girls, few significant CPOX4 Hg interactions or independent main effects of drugs were observed. In contrast, among boys, numerous significant interactions effects between CPOX4 and Hg were observed, spanning all 5 domains of neural behavioral performance. Now, if I can read that, and it's telling me that mercury affects boys more than girls, especially in neural behavioral performance, why would you not think that exposing boys to thimerosal at high levels, what would be EPA safe if they weighed 275 pounds, as I pointed out years ago, that this would not be something we should eliminate and stop? And why would you suggest that maybe we should give pregnant mothers a flu vaccine? I mean, there is a lack of intelligence in the CDC that I find phenomenal. And I don't think you can be nice to these people. They're not people that respond to being nice to them. You have to tell them, I've lost all respect for you, and I think Dr. Wakefield's presentation just adds to that. They're administrators. They're not scientists. I mean, I would like to see the papers that these people have ever published. Have they ever worked in a laboratory? What were their biochemistry grades? They obviously didn't do very well. So, and just to summarize, just to say, these findings are the first to demonstrate genetic susceptibility to the adverse neurobehavioral effects of mercury exposure in children. I don't think they were the first, but they definitely demonstrate that. And since then, he has now published 3 or 4 more papers that follow up. And what it says, he has to admit to himself, he missed this when he was doing the work with the dental people at the University of Washington. And all of us need, the production of these papers now by totally different science, by Dr. Woods, is based primarily on Wouldn't have happened if the Geyers hadn't done and published their paper on showing that he mishandled the way he did his statistical analysis in the cat study. But I think there's a question here, and I got it down here at the bottom to remind myself. Why is the effect only statistically observed in boys? The testosterone may contribute by increasing the uptake of mercury as suggested by the lower urinary excretion of mercury. It is not the presence of the CPOX4 gene alone. So when we look at people that get sick, it's not simple. I mean, and I hate to say this, it's just incredibly complex. But you have the genetics, plus you have the gender. So if testosterone increases your retention, and then you have a genetic susceptibility, you're hit. If you don't have testosterone and you don't incorporate so much or maintain so much, like a girl would not do, you don't get sick. So we need to somehow make Congress understand that the people that run the vaccine program have to be more intelligent than what they've shown themselves to be, or at least capable of logic. Okay, this is where I talked to you about this. I'll show you the data now. And this is how mercury compounds concentrate in cells about 5.6-fold higher than in the blood and in the mitochondria by over 1,000. This is done by Dr. Martin Sharp in toxicology. And I know this will drive you crazy, so I'm just putting it up to show you. I have read this and I've studied it, and he's exactly right. And what you see here on the first part in this region here or here is that it's the membrane potential that gives you the 5.6-fold increase or the 1,000-fold increase, because you can use an equation called the Nernst equation and determine if things came to equilibrium how much they would concentrate across a membrane that has different levels of negative charges on each side of the membrane. It's biophysics. I happen to love it. A lot of people makes them throw up, but you have to be weird to do this kind of science, and I think I was weird from the time I was born. But anyway, just if you want to read this, and anybody that's in here that has doubts about this, that has a PhD and wants to educate themselves to a high level, it's Martin Sharp. It's in the Journal of Toxicology in 2012. And what he showed is that thimerosal from vaccines, while you're talking about the amount in the blood, and if you remember, there were several papers sponsored by the CDC that said, well, it leaves your blood too fast to be toxic. Well, hell yes, it leaves your blood fast because it's going into your mitochondria. And ethyl mercury goes out of the blood faster than methyl mercury because of the Nernst equation and the type of chemistry that's involved. And so when something leaves your blood, that doesn't mean it's not toxic. And that is, I want to tell you, to a scientist, that tells me that the people that did that study, which they tout them as being really intelligent, they're not intelligent at all. They've missed fundamental science. I mean, the alcohol that makes you drunk isn't the alcohol that's in your bloodstream. It's the alcohol that gets in your brain. And it goes out and it gets into your body very quickly. And before you ever get it out, all of it out of your blood, it's concentrated in your brain to where you can't even speak, like I'm talking today. And I apologize for garbling some words, but I can't help it. And this is a take-home for all you guys that treat people for different diseases, especially for mercury toxicity. Mercury, Hg2+, cannot directly make free radicals. It will not give up another electron or donate an electron to something like a hydroxy radical, to perform a hydroxy radical. And this is done by Fe2+ or Fe3+, or copper or manganese, that's displaced in a cycling process using iron displaced by mercury, which binds tighter to the sulfur binding sites, the sulfur iron centers in the electron transport system of the mitochondria. So if you're going to treat someone with chelation, you've got to address the iron initially, because there's a lot of iron that's been released by the mercury that they have inhaled, that's been converted to Hg2+, that goes in and binds to these enzymes. And this is the Fenton reaction for all of it at once. And what I'm telling you is that it takes mercury cannot do this. Copper can do this. Manganese can do this. But those are the other two natural occurring metals that might be involved. But it's only when you release these Fe2+ from the electron transport system of the mitochondria, then it starts converting and making the Fenton or the hydroxy free radicals. And this is the Nernst equation for those of you that want to memorize that. quickly, just I'll give you a sec. But you use this and you can calculate how certain things would concentrate on different sides of the membrane. At least this is how Dr. Sharp did this, and say that in biophysical chemistry, that's a very important equation. And the membrane, if you look at cell membrane is 45 millivolts, and the mitochondrial membrane is around 80 millivolts. So it concentrates more in the mitochondria after it gets inside the cell. And this tells us how one mercury, or one ethyl mercury atom, can produce a huge number of hydroxy radicals, causing massive destruction. Again, this is the same thing, and I'm not going to sit here and the slides are out here. I put this on because I know you get to copy the slides, and if you want to see if I'm being honest, or if anybody at the CDC wants to hire somebody that's intelligent enough to read this, they can have it confirmed, or they can find holes in it if they'd like. And when you start producing these hydroxy-free radicals, you consume all the glutathione, because that's the main source of removal of hydroxy-free radicals from your body is the glutathione. Give me a warning. 10 minutes? Okay. Okay, now this is something I thought about. Is oxidative stress related to illness? And this is how you can test this. You go to Google and pick out your favorite disease and type the name down. And they say, and oxidative stress. You will be hard-pressed to find an illness that is not associated with oxidative stress being developed in the tissue you're talking about. And so what causes this? I mean, and then you have to say, what can get in and out? Trust me, I believe in antibiotics, and I know about bacterial infections. There are a lot of things that can make you sick besides mercury. But basically, when they get on, what they're doing is they're making iron, releasing your iron, because if you don't have iron-free iron, you cannot have hydroxy radicals and inflammation without some metal donating electrons in a catalytic sense to the oxygen that's in your body. And when I've done this a long time ago, it says that every neurological illness is associated with oxidative stress in the brain. And that means there has to be a metal in the brain that's making hydroxy-free radicals, and the one that could be is either iron or copper. It could be manganese also, but the most likely ones. And here's some papers just to show you that I do read some stuff and I collect some stuff. This is effect of mercury dental amalgam fillings on renal and oxidative stress biomarkers in children. And it says, our data provide evidence that low exposure to mercury from dental amalgam fillings exerts an effect on kidney tubular functions in children. Oxidative stress may have played a role in this mechanism. My God, I mean, be definite about things. There's no doubt. I mean, if you look at it and say, what else could do it? And what this is talking about, amalgam fillings have an effect on the kidneys of children. So you don't have to, you can use amalgam fillings if you want, if you don't care if your child is going to operate at maximum efficiency and at maximum ability. And the CDC can look at this kind of data, and the FDA, and just totally ignore it. I mean, they totally ignore it. How many years has it been since we shoved it down their throats? And even the outside advisory committee to the FDA said, you haven't proven that this stuff is safe. And what they really observed in that meeting is that they cherry-picked the papers that they put out, all written by dentists, and some of them not even scientific papers. So we have a government, and this is really the problem. And I would tell you, when you call your congressman to help Andy out, that you also tell those a bunch of SOBs that you're killing our kids. I mean, how can you explain that we vaccinate earlier and more often than any other country in the world, and yet we have one of the highest infant death rates in the world and one of the highest childhood death rates? If that doesn't make you mad, and I'm not talking about that today because I don't feel like it. I don't need the extra adrenaline pulling out, because that gets me. What's wrong with our country? There's something. You can't say we have a great country if we can't address that problem, when it's not a problem. When we have people dying of sudden cardiac arrest, not a heart attack, their heart just stops beating, and most of them, or a good number of them, are young male athletes playing basketball or running track or doing something like that, and they have this high mercury level in their heart. And people say, well, you don't know what causes it. What do you mean you don't know what causes it? You won't if you don't look. And that's what they don't want to do. They don't want to look. And the same thing with the amalgam fillings in children. We are damaging, and we have successfully damaged our children for a long time. And I would tell you also, I think giving a mumps, measles, and rubella vaccine to a child that has had his intestines disrupted by thimerosal would be an exceptionally bad idea. Okay, this is something I just thought this would be important. It says this is a study from the Catholic University of Louvain, done just recently, and it said metal ion homeostasis and its dysfunction, which results in increased accumulation in brain, notably of copper, iron, and zinc, may be associated with a number of neurological diseases, such that chelation therapy may be the one therapeutic option. Now, to have the therapeutic option and the chelation work, it has to cross the blood-brain barrier. But you also have to look at this and say, why is the accumulation of copper, iron, and zinc in the brain increasing? How does it do that? I mean, it's almost impossible for you to take, even if you injected it into your bloodstream, iron, and have that iron get into the brain. I mean, there's just too many protective factors there to stop that. There's too many iron binding proteins, too many chelators that would bind the iron that would prevent that from happening. And I'm telling you that the iron and zinc and copper that increase in the brain that they see associated with Alzheimer's, Parkinson's, and other things, got there through natural processes, healthy processes. It's after it gets in there and then you get exposed to something like cadmium or mercury, which mercury being the main one because it goes in very easy, you can breathe it, and then it gets released in situ at the site where it causes the damage. So you have to ask the question, why do free essential metals build up in the central nervous system? Because they can't get out. I mean, they go in there, they're carried in there by iron-carrying proteins, for example. They get put onto the electron transport system and the iron sulfur centers, which is the highest level of iron in your body. And then you take mercury in there, and mercury knocks the iron and the copper off of their nitro binding sites, releases Fe2 plus and copper, and they can't go out. The blood-brain barrier will prevent iron from being excreted from your brain. I think that's been fairly well established a long time ago. Okay, this talking about oxidative stress, and one of the key things, apoptosis, and I want to make that on the same, is controlled by the ratio of GSSG. When your GSG levels go off, your electron transport system gets shut down, and that cell dies. That's to get rid of tissues that are supposed to die during development, such as a lady who has had breastfeeding a baby, and now she quits and she doesn't have a baby to feed anymore. Those cells will atrophy. And it's done by this normal process called apoptosis. But if it's done by the ratio, and almost everyone will agree it's done by the ratio of these two, then if you abnormally induce a ratio that's odd by causing oxidative stress in the mitochondria, then cells that should not be dying will die of the same system. I mean, they will lose their electron transport system capability. Okay, this is and I'll finish this very quickly. I'm going to show you how we've developed a compound. And this hasn't been easy, folks. I got more arrows in my back from people who want to keep people sick, in my opinion, and it's real simple. I applied farm boy logic to the process. How do you get toxins out of the body that you can't keep from getting in there at some level? Mercury, primarily, cadmium, and the effect of iron. And so we made a lipid-soluble membrane or chelator that can penetrate cell membranes with ease. It also crosses the blood-brain barrier and enters the bone marrow. This is all published. It's all been proven. It is without significant toxicity, either taken orally, injected subcutaneously at very high levels. And this is the NBMI with a CTI. It does not cause a depletion of essential metals. It binds mercury and renders it non-toxic, even when the cells and tissues of the body contain high levels. We made the mercury NBMI complex. I made it outside the cell. And we gave 2,000 milligrams per kilogram body weight to rats, injected it down their throats. And they excreted all of it, and they didn't show any signs of toxicity, even though the mercury was, they had to go through their digestive system, got into the body. The NBMI mercury complex is without toxicity, and it doesn't break down even if you feed it to animals or inject it into them. And this compound effectively scavenges also, in addition to binding the metals that make the hydroxy radicals, it effectively scavenges the hydroxy radicals made by mercury, toxicity, and other toxic effect. It can be taken orally and clears the body in test animals by about 90% within 24 hours. But since then, we've done the human studies, what they call the phase 1, and this is a copy of the molecule, a structure of it, and it passed the phase 1 without a single adverse effect. And this is giving 600 milligrams a day for 10 days in a row. And what this compound is, if you look at it, it's made of two compounds, dicarboxybenzoate and cysteamine. And this is the reason, I was really enthralled with the molecule when I first met it, is that it's made of two natural products. This is found in cranberries and apples, and this is found in meat. And it's on the terminal end of coenzyme A. And it's used in medicine or in dietary products to prevent oxidative damage, like spoiling. So this compound, the fact that it's not toxic isn't an accident. And I would point out to those of you that are very chemically involved that if you look at the benzene ring part here, this is a site where the P450 system can attach a hydroxyl group, and then you can attach a glutathione to that to carry out anything it's bound to that it doesn't want to let go of. So it has designed into it several advantages, and it binds mercury incredibly tightly. And once it binds mercury, it never lets go. We can't do anything except actually fire and destroy the compound's structure to get it to release mercury. And if you talk about it in metals, how many metals it binds, this showing is that mercury, lead, cadmium, copper, and iron are all very, very tightly bound with this. You can take all the metals out of water by exposing it to this compound. And so the same thing would happen in your body if you could get it there. And we can get it there. So this is something that's up and coming, and we're making progress on it. It doesn't leach. It never lets go. You can see the leaching process for a long time is very, very low. So once it binds these metals, it doesn't let them go. It renders them non-toxic, because if you cover up the electron shell, they can't donate electrons and make more hydroxy radicals. And I can go through this, but I think it's the best, because I only got a few minutes left. We've done every kind of thing. I have injected this into rats at such high levels, I was feeling sorry for the rats. But when I looked at them, the ones that got the highest levels were the ones that looked the healthiest. It didn't bother them at all. And so we know this compound is very, very low toxicity, and it's going to be something that's going to be very effective and helpful for people. And I have to take this through the orphan drug program of both the FDA and the EMA, and we're making good progress, but it's slow. I am not a patient type person. I hate regulatory aspects when they're nonsensical. And the problem here being is if you take a drug and you give it to a rat and a milligram kills him, you have to do, and you have another drug and you give it to him and you give him 5 grams and it doesn't bother him at all. You still have to go through the same safety testing for the one that's really toxic versus the one that's non-toxic. And so we have to do everything they say, because they're the 800-pound gorilla. And I would tell you up front, it's not available. We can't give it to you, and it's not that we don't want to. But if the FDA saw me giving you some of this compound to treat your ailments, they would use that to destroy the company and me, and I can't take it. Okay. The other thing about it is its antioxidant properties, and I just want to jump to this. This is a NORAC score, oxygen radical absorbance capacity test, and the top one is the NBMI, or OSR as we did it at that time, and it's an incredible hydroxy-free radical scavenger. There's nothing comes close to it, and it's not toxic. You can take it orally, you can inject it. I think you could do anything with it, and so I think we have found a very good way that we're going to be able to help people that are sick as soon as we get this through the FDA's and the EMA's thing. And you say, how does it do it? If you take this is a study done a long time ago by Dr. Julie Buckley. We did it on 10 people. These are some of the individuals. And you can see from the lowest level, if you look at the GSH level up to here, and just and every one of these people, 10 out of 10 people that took this, had a tremendous increase in glutathione levels and reduced glutathione levels in their body. And I'm 70, I'll be 74 this month, and my birthday's not very far away, so if you want, I'll give you my address. You can send me a birthday gift. Okay. But when I have my glutathione levels checked, they were 1280. People my age are usually around 200. So this does build up the glutathione levels in people in this food safety testing that we've done. So it's very effective. And it's not added. This compound does not add to anything in your body. It just scavenges the hydroxy-free radicals, keeping you from consuming what you normally make. And I think the value of this compound is going to be is that it's not going to replace medicine. But if a doctor's treating you with good nutrition, etc., this is just going to make it work better. That's all it's going to do, because it doesn't do anything. It doesn't repair any of your membranes. It just prevents you from doing the damage by producing hydroxy-free radicals. And as one of the earlier people talked about yesterday, Chris brought up the fact glutathione S transferase is the enzyme that transfers glutathione to sick people, to toxins, to get them out of your body. And when we tested these people, who were all sick, by the way, the level was low to 0 for the first, and it didn't rapidly turn around. But at the end of the study, every one of them had increased glutathione S transferase. But this is an enzyme that uses glutathione as a substrate to mark toxins for removal from the body. So it's another good thing. So the conclusions are we have a non-toxic, lipid-soluble, free radical scavenging antioxidant with mercury. And I used to dream about this, about being able to do this. You know, it tells you that dreams can be accomplished just with hard work and thick skin. You have to have the thick skin, trust me, because I can't tell you how many critiques I've had on this molecule before I ever got a chance to show it wasn't doing it. And they could have been good. They could have been right. I mean, you have to test things. We've tested this beyond belief. I mean, roughly several $1 million worth of testing, and that's what they require for a dietary ingredient. I mean, I can't tell you how I feel about that. And so I think I'll end here. I think that's probably the best place to put an end to it. Thank you.